installment of our 2015 webinar series, and we're thrilled to have with us Dr. Dane Cook as our presenter. Just a little bit about Dr. Cook. I think he's very familiar to many of you. He's the co-director of the Exercise Psychology Lab at the University of Wisconsin-Madison and director of the Marsh Center for Research in Exercise and Movement. Dr. Cook received his master's and doctoral degrees at the University of Georgia and postdoctoral training in neuroscience at the University of Medicine and Dentistry in Newark, New Jersey. You know, his research really focuses on the psychobiology exercise with a specific focus on how exercise influences the central nervous system in both health and in disease. Recently, Dr. Cook's research has begun to incorporate additional biological systems such as the immune, autonomic, and gut microbiome to better understand how distinct yet related physiological responses interact to maintain illness. His work in MECFS has demonstrated an augmented brain response during cognitive processing, altered cardiopulmonary responses to submaximal exercise, and augmented gene expression and symptom responses to maximal exercise. Dr. Cook was recently awarded a 5-year Merit Review Research Award to test the consequences of acute exercise on the brain, immune, and autonomic nervous system function of Gulf War veterans, and we know that that will have spillover effect for our illness. Dr. Cook's research has been funded by the NIH, the Department of Defense, the Department of Veterans Affairs, and of course we at the Solve MECFS initiative have been very proud to fund Dr. Cook's research. He also serves on the Chronic Fatigue Syndrome Advisory Committee, the CFSAC of the Department of Health and Human Services. So with that, I know Dr. Cook has a lot to cover, so Dane, take it away.
Thank you, Carol, and good afternoon, everyone. Morning if you're on the West Coast. Very happy to, again, talk about our research looking into post-exertion malaise, which to date has all been funded by the Solve MECFS initiative. So I assume I have control now. Okay, so the title of my talk is Deciphering Post-Exertion Malaise, really looking at the intersection of biology and behavior. For today's discussion, I'm going to talk about the clinical and laboratory characteristics of post-exertion malaise. So by clinical, I mean looking at the patient perspective of it, the research case definition perspective of it, and then juxtaposing that with how we study post-exertion malaise in the laboratory. I'm then going to talk about our preliminary work where we are studying post-exertion malaise. And then I am going to get into the detail about that work. And then I'm going to end the presentation with exploring some of the individual differences that we see in post-exertion malaise. So, oops, sorry about that. So a lot of people, I think, are asking now, what is post-exertion malaise? And this is quite a change from research a decade ago where we were just really doing the research to establish that it was present. In 2010, Dr. Anthony Komaroff called post-exertion malaise an illness within an illness. And I think that this is an interesting characterization of PEM, the idea that patients are trapped within their bodies and, for that matter, within their brains, and attempts to improve their quality of life by either shopping, walking the dog, or trying to play with their children can dramatically worsen their condition. And indeed, now post-exertion malaise has become a defining or cardinal feature of MECFS. But I think it's also a term that doesn't adequately describe the magnitude of illness change that a patient can experience when they exert themselves. When you look at the patient perspective and look at personal testimonies, post-exertion malaise is described as a debilitating crash following minimal exertion that can take weeks to recover from, An example such as the patient would need to stop grocery shopping mid-shop, stow their shopping cart with their belongings in it, lay down in the car for a half an hour, and then when they feel like they're able to, return to finish shopping. Others have described it as completely unable to perform activities of daily living, the loss of ability to think, a roaring within their ears, loss of balance, rooms turning upside down, and even poison oozing from their body. Here on the left we have an artistic rendition by Michael James Hawke of mental exhaustion, where he describes the lack of harmony between the physical and mental self, what I call a psychobiological chaos. So here we have the arms and the torso and the legs and the head. And this piece struck me because it illustrates the lack of harmony between the body and the brain during an exhaustion state, how the physical self battles the mental self. And to me, this captures many of the patient descriptions of post-exertion malaise. A patient tries to be active, to participate in life, and it is a battle between body and brain, and this leads to both physical and mental exhaustion, causing them to crash. So when we look at the research and clinical case definitions, from the research case definition perspective, post-exertion malaise is described as extreme prolonged exhaustion and sickness following physical and mental activity, lasting more than 24 hours. When we look at the clinical case definition by Carruthers and colleagues, it's described as debilitating malaise and/or fatigue, generalized pain, deterioration of cognitive functions, and worsening of other symptoms occurring either immediately or a delayed period of time following physical or mental exertion. So you can see even within the case definitions for MECFS, we have variability in how post-exertion malaise is described. Really the idea that of post-exertion malaise rose to prominence even more with the publication of the IOM report this past spring. And this report went as far as to propose a name change that focused on this debilitating condition. But I think more importantly, it resulted in the single most comprehensive and informative review of the literature that we have to date. And within this report is evidence that demonstrates that this disease is real, it's debilitating, and that I think from a research perspective, at least from my perspective, that it impacts multiple biological systems and multiple physiological processes. And here's the illustration from the IOM report of the percentage of MECFS patients and healthy controls, the patients being in the blue-green bars and the controls being in the orange, reporting post-exertion malaise symptoms of at least moderate severity at least half the time during the past 6 months. And so you can see the vast, vast majority of patients are reporting this phenomenon. Here are some of the key outcomes from the IOM report. I think really importantly that MECFS is a serious, chronic, complex, multi-system disease that frequently and dramatically limits the activity of affected patients. They proposed that new diagnostic criteria were needed that simplified the diagnostic process but really need to be validated. And they went as far as to say a new name is warranted because they felt that the review made it clear to them that the term CFS was dismissive and damaging to patients, but that the term ME was not an accurate description of the literature that they reviewed. Here are the diagnostic criteria. Again, just showing here that you need a substantial reduction or impairment of the ability to engage in pre-illness level activity, bringing post-exertion malaise in prominence as a required criteria, as well as unrefreshing sleep, and then at least one of the following of either cognitive impairment or orthostatic intolerance. And I want to highlight the asterisks, which I think is often overlooked, that the frequency and severity of symptoms should be assessed, and that the diagnosis of MECFS should be questioned if patients do not have these symptoms at least half of the time with moderate, substantial, or severe intensity. And so these new diagnostic criteria, although they're simplified, still include both frequency and intensity components. And this needs to be educated to the physicians. So this kind of brings us back to the question, what is post-exertion malaise? So if you look at clinical descriptions and personal experiences and the diagnostic criteria, these all characterize post-exertion malaise as a debilitating exacerbation of the entire constellation or gestalt of what MECFS is. But within these, we see lots of variability in its time course, the severity of the symptoms, and the types of symptoms that patients are reporting. And this is the area that really needs to be studied so that we have good baseline knowledge of what PEM is. So this brings us to what do we see in the laboratory? Well, the typical lab scenario would be that you'll have your outcome or your dependent variable of interest. You will measure that at baseline when a patient is resting, and then you will give them a stressor such as acute exercise, have them do that single bout of exercise, and look at how that impacts that variable that you measured at baseline. And it turns out that post-exertion malaise in the lab is largely defined by what the expertise of the investigator is. So for example, many studies will report that post-exertion malaise is an increase in symptoms such as pain and fatigue. But there have been few studies that have really measured the entire construct or symptom constellation that makes up PEM. Other investigators have operationalized it as a reduction in physical activity levels following an acute bout of exercise. Others have characterized it as abnormal exercise responses. This is particularly true in the back-to-back maximal exercise testing procedure, where a patient comes in, they do a maximal exercise test, they are asked to come back 24 hours later, and they look at the metabolic responses again to demonstrate that patients are unable to reproduce the same level of performance that they did in the first exercise test. And so they're uncovering metabolic abnormalities there. Still others have operationalized it as a change in cognitive function post-acute exercise, or a change in their sleep or their circadian regulation. Impaired pain regulation has also been studied under the scope of PEM, and perhaps the most widely studied are these various biological markers. So whether they be cytokines, other measures of immune system, or oxidative stress, they measure these markers at baseline, they stress the system with acute exercise, and then they see how these various biological markers change. So you can see the variety of outcomes that have been used to characterize post-exertion malaise. And what this really points to is that this phenomenon likely involves multiple physiological systems. And so this this really kind of leads to where my lab, the direction my lab is taking. We have these multiple physiological networks that are designed to interact and be adaptive to stressors when they're placed upon them. What we hypothesize is that in MECFS, post-exertion malaise detrimentally impacts many of these systems. So what we want to do is take this network physiological approach, examine multiple systems within the same individual, and determine the deficits or breakdowns that occur in those systems when they are stressed. Then we want to determine how these systems interact to produce, maintain, or worsen symptoms. And our real long-term goal, and we're just at the beginning here, is to then use this information to create novel treatment approaches that are based on targeting these physiological deficits or breakdowns. So this brings me to the research that we're currently doing. And my interest and start to one moment, please. The slide is not advancing. Okay, there we go. So I really got back into MECFS research with this study that was conducted by my master's student Jacob Meyer in collaboration with Dr. Sanjay Shukla at the Marshfield Clinics and Dr. Alan Light at the University of Utah. And it was really based on Dr. Light's first study that he published in 2008 showing that there was an increase in gene expression following submaximal exercise. So exercise at 70% of a person's peak capacity. What we wanted to do in this study was to extend upon that and use maximal exercise test to address some of the concerns that were raised by the initial study. And so the purpose of this study was to determine gene expression and symptom changes to a standardized maximal exercise protocol in patients with MECFS and in healthy controls. So here is a brief overview of our study protocol. So on the first day, we brought them in for a history and physical, did a clinical interview, and we collected both blood and stool samples and baseline symptoms. We then brought them back about a week later for their second day of testing, where we had them undergo maximal exercise testing to exhaustion. And prior to this test and after, we collected symptoms and blood samples. We then brought them back 48 hours later, assessed their symptoms, took a stool sample and a blood sample again, and then brought them back a third day, 72 hours later, to do those same measurements, to look at the time course of post-exertion malaise as well as gene expression. So the extension here is we're using maximal exercise testing instead of submax, and we're taking it out to 72 hours. And so here we have our gene expression results from that first study that we published. So on the y-axis here, we have the increase in their gene expression, mNRA. On the left, we have our controls, and on the right, we have our patients. And you can see here, based on these bars, that we have sustained increases up to 72 hours post-exercise for adrenergic receptors, so those that are involved in energy production, and for the NR3C1 receptor, which is our cortisol receptor. And so we're having an increase in gene expression for these two proteins or receptors that we're not seeing in healthy controls. And at the same time, we're also seeing an increase in symptoms. So here we have the change in symptoms from baseline. Again, the controls are on the left, and the CFS patients are on the right. And here we're having sustained increases in confusion, pain, and fatigue that we see in the CFS patients that mostly resolve by 72 hours post-exercise. And in controls, we're seeing an initial immediate increase in pain, but then after exercise, they tend to get better, not worse. And so this is a demonstration that we're having changes in gene expression. We're having changes in symptoms in our MECFS patients. And when we look at the relationship between the two, we see significant positive correlation. So here we have the change in the cortisol receptor from baseline to 48 hours post-exercise, and the change in a single symptom here of fatigue. And what we see here is that those patients who had the greatest change in their fatigue also had the greatest change in the cortisol receptor. And this is really important to demonstrate that we both have a change in biology and in a change in illness, because we need those two changes together to be able to describe the phenomenon of post-exertion malaise. So that study led to our next funded study by the Solve MECFS initiative to really extend our research and add in the brain. And so here we have post-exertion malaise in MECFS, brain inflammation, and behavioral interactions. So we're doing essentially the same type of study that we've done in our maximal exercise study that I just presented to you, but now we're adding in neuroimaging to look at brain activity. And so this is really a collaboration. Again, we're extending our collaboration with Alan and Kathy Light, and we're bringing in Gordon Broderick at Nova Southeastern University to take more of a systems biology approach to the study of post-exertion malaise. So we're collecting blood to look at the immune system, and we're looking at the brain as it performs cognitive tasks, which I'll describe in a moment. We're stressing the system with exercise, and then we're taking these measures again. So the overall goal here is to determine the dynamic relationships between brain structure and function, gene expression, and symptoms in MECFS patients, and we're using exercise as a model of post-exertion malaise. Here again, here's our study protocol. So on the first day, we do a detailed screening of our individuals coming into the study. We then bring them in for baseline testing where we get detailed symptom information. We collect blood, and we do our first round of functional magnetic resonance imaging or brain data collection. And for this, we have while the patients are lying within the magnet, we have them perform fatiguing and non-fatiguing tasks. For the non-fatiguing task, we have them perform finger tapping so that we can look at motor cortex activity. And then we also have them do a non-fatiguing cognitive task where every time they hear the number 7, they press a button. We then have them perform a fatiguing cognitive task, and this is called the paced auditory serial addition task. So here they're listening to a series of numbers from 1 to 10, and every time 2 numbers add up to 10, they press a button. So for example, they'd hear the number 8, they would hold that in short-term memory. They hear the number 1, and they do nothing because that does not equal 10. They hold the number 1 in short-term memory. They hear the number 9, 1 plus 9 equals 10, they press the button. Now they hold the number 9 in short-term memory, and they add it to the next number. And we have them do this for 12 minutes. And we've shown in several studies now that this induces mental fatigue. After they do these baseline tests, we then have them come back a week later where we collect blood, symptoms, and then have them perform submaximal exercise at 70% of their heart rate reserve. We collect metabolics during this, heart rate, blood pressure, and we take lactate samples. And then after exercise, we collect blood and symptoms again. They come back 24 hours later, and they repeat day 2, where we collect blood, symptoms, and have them do our brain imaging tasks. So that is an overview of our study protocol. So let's get into the results. So here we have the symptom changes that occurred from baseline to 24 hours post-exercise. And what we have here on the y-axis is the change in these symptoms for 10 visual analog scales that range from 0 to 100 of the CDC inventory. And so we have 10 symptoms that are aggregated. The controls are in black, and the MECFS patients are in red. And as you can see, there's a dramatic worsening of symptoms pre to 24 hours post-exercise for patients, while controls get moderately better pre to post-exercise. So the first thing has been established. We've seen a change in the symptoms. Our patients are getting worse. Next, we looked at their cognitive performance. So here we have the total number of errors that occurred during our fatiguing cognitive task that I just described to you out of 135 trials. We have 3 blocks, both pre-exercise on the left here and 3 blocks post-exercise on the right. The CFS patients are in red. The controls are in gray. And as you can see, they start out the same during that first block of cognitive testing. CFS patients and controls are the same. The controls tend to do better over time. They come back 24 hours later and they get even better. And what you see in the patients is the complete opposite response. As they become more mentally fatigued, their cognitive performance worsens. They come back 24 hours later, they start off worse than they began the day before, and they continue to make more errors throughout the 12-minute test. So we're having an opposite response. We have an increase in symptoms in our MECFS patients but not controls, and we also have a worsening of their cognitive performance that we don't see in controls. So what is happening in the brain while all this is occurring? So here we have a series of images that we collected at baseline during the fatiguing cognitive task. Not pictured here are that there were no differences in the brain activity between patients and controls for our finger tapping task or for our simple cognitive task that didn't induce fatigue. But when we had them perform the fatiguing cognitive task, patients had greater brain activity in the anterior cingulate cortex, bilateral hippocampus, and the dorsolateral prefrontal cortex. All of these brain regions are involved in executive function tasks like the one we are asking them to do. The fact that patients are showing a greater amount of brain activity in these tasks compared to controls suggests that they are having to use greater neural resources to complete the cognitive task, and they're still committing more errors. They are also showing less activity in this superior and inferior parietal cortices, and these are involved in sustained attention. This suggests that the patients are having a harder time sustaining attention on the task than our healthy controls. And this is all occurring just at baseline, before we even use exercise. These are the responses to exercise. So not pictured here is that the controls, when they got better at each one of these tasks, pre to 24 hours post-exercise, they actually had less brain activity from pre to post-exercise. In patients, what we saw was an increase in activity in some of these same brain regions. The frontal cortex, here the dorsolateral prefrontal cortex, which is involved in executive function, the hippocampus, which is involved in memory, and the cingulate cortex, which is involved in filtering of the information. So again, we're seeing now the effects of exercise is a worsening or an increase in the neural response to fatiguing cognition. And importantly, the changes in these brain responses are also related to the changes in the symptoms that we're seeing. And so here is one example where we looked at the relationship between the change in their feelings of fatigue and their brain activity during the fatiguing cognitive task. Those with greater fatigue were showing greater activity in the parietal cortex, and there was a negative relationship in the posterior cingulate cortex. So to summarize, we're having a dramatic increase in symptoms, we're having a worsening of cognitive performance, and this is reflected in their brain responses. So now we have objective brain evidence showing the consequences of acute exercise on these patients. But I don't think that that's the entire story, because now that's presenting to you the group data, which is, I think, extremely important. But the next thing that we're doing is we're starting to look at the individual differences that are occurring among patients and controls. And I think this is important. If we're to truly understand post-exertion malaise, we need to know for whom it is most prominent for, what symptoms are changing the most, the variability in those symptoms, and the variability in the biology that is occurring as a result of the exercise stressor. And so I went back and I started to look more individually at our data. And so let's start with the symptoms. So here's what I showed you just moments ago, the worsening of symptoms in our MECFS patients from pre to 24 hours post submaximal exercise. Here's the variability in those symptom responses. So here we have the change in symptoms from pre to post exercise on the y-axis for all 10 of the symptoms we measured on top of one another for each patient that we measured it in. So we have the bars are all the symptoms on top of each other, and each number here is an individual patient. So I think it's very clear the amount of variability we're seeing in the changes, at least, of their symptoms. We have some people who are having dramatic changes in their symptoms from pre to 24 hours post, some who are having small changes in their symptoms or relatively small, and some who are changing very little or even getting somewhat better. I think we have to start to begin to understand why this variability is occurring. One of the things that we have to keep in mind is from where are the patients starting when they come into the lab? Are they having a good day or a bad day? And so here again is that same graph, the change from pre to post exercise. And right here on top, we see where they started. These are their symptoms at baseline. So for example, here is a participant that came in who was very sick. They were rating very high on all of our scales. This means that they had very little room to move from pre to post exercise, and that is exactly what happened. This is not because they're not feeling sick. This is because our scales are not sensitive enough or detailed enough to pick up the changes that they're feeling. Or they're so sick they have so very little room to move. Here, on the other hand, is a patient who came in and was maybe having a better day for them. 24 hours post-exercise, they had a large change in many symptoms. That is, I think, typically what we think of in post-exertion malaise is this large change. And then another example would be a participant that came in who was very ill on the baseline day, and whatever amount they could move on our scales, they did 24 hours post-exercise. So not only did they come in and have severe symptoms at baseline, they also had a large overall change in number, in their frequency, and severity of their symptoms. So this is the type of variability in post-exertion malaise, along with other things, I think, that we need to begin to look at. So this also got me thinking about our other dependent variables. Well, what about the cognitive performance? So here's the cognitive performance that I just showed you earlier. CFS patients getting worse over time while the controls are getting better. So what we did here was we looked at the individual differences or the variability in our cognitive performance. So on the y-axis we have the total number of errors again. Along the x-axis we have participants who had large changes in their symptoms, participants who actually had a small decrease in their symptoms, and then controls. And what you see here is those who had high ratings for post-exertion malaise made the most errors, while those who had a small decrease in their symptoms either made few errors or actually got a little bit better. And then these are our three worst performing controls. So even within our cognitive performance, we have individual variability that we need to consider. So this means that there might also be individual variability in biological responses. And we haven't gotten totally into our gene expression data from our most recent work, so I went back to the study that was conducted by Meyer et al. And started to look more individually at the gene expression data. So here again we have the sustained increase in the adrenergic receptor and the cortisol receptor in our CFS patients that was not present in controls. And when I looked at the gene expression data on an individual basis and related it to symptoms, it didn't hold up quite as well as our cognitive or our symptom data. And again, these are just examples and we have a small sample, but I think it's really telling and informative. So here we have the symptom changes on the X, and we see for someone who had improved symptoms, someone who had a large change and someone who had a moderate change, we have large changes in their gene expression, particularly for the AD2A receptor for these two individuals. But here we have an individual who had one of our largest change in symptoms from our first study, who, relatively speaking, had a moderate change in their gene expression. And here are individuals, moderate changes, who had improvements in their symptoms. So even though gene expression was changing in the positive direction for everybody, it did not appear to map with symptoms as well as our other two outcomes. So I think it's clear from the literature, from our own research, that post-exertion malaise impacts multiple physiological systems. We now have to begin to determine how these systems are interacting with one another and how these interactions explain why patients get more sick. And then hopefully this will lead us to being able to create treatments that are aimed at addressing physiology or biology. I think there's also a large gap in our baseline knowledge of what actually post-exertion malaise is. What is its time course? How variable is it from one person to another? How severe can it get? And what are the primary symptoms that are reported? Looking at these individual differences, I think, is important to help us answer this question more thoroughly. And with that, I thank you very much for your time and attention.
Dr. Cook, thank you so much. This is Carol Head again, and we have quite a few questions from the group of folks on who are watching the webinar. So I'll start you off, and we'll see how many we can get to. I believe we have about 20 minutes to cover questions. So first up is a question about some of the specifics of your methodology.
Sure.
What do people do during the 48 and 72-hour intervals? Are they on bed rest or limited activity? What are they doing?
So for our studies, these first two at least, they went home and they reported that they did various things. Some went about their daily activities. Some reported that they crashed and were in bed. Some reported that they felt worse, but that they tried to continue what they would normally do. So we do not control what they do at home, at least at this point.
Mm-hmm. Okay. And then similarly related about methodology, do you take into consideration whether the CFS patient is in remission or relapse at the time they enter the studies?
Yes, we do. And within our screening, they have to meet current criteria. So they cannot be in remission or relapse to qualify to be in the study.
Okay. Okay. All right. Next up. Can you comment on mitochondrial disease / dysfunction in MECFS?
So this is currently a hot topic of research, not my expertise. The idea here is that the mitochondria, which is basically our engine for energy within our body, that they are either somehow dysregulated within MECFS and in related illnesses like Gulf War illness, and because they become dysregulated, they're not able to produce the engine isn't able to run and produce the energy that we need in an efficient fashion, and over time this creates a vicious cycle. Where exactly the breakdown is in the mitochondria and the exact biology that underlies it, I'm not clear on. But that's the idea, is that this is our powerhouse for energy within our body, and that if we have a breakdown here, it would make sense that you would be able to sense that and that you would feel ill.
Thank you. Thank you. Next up. I find that when I feel not like my 100% self, if I lie in the supine position for a while, the symptoms go away faster. And after about a couple of hours later, I feel better and can be more functional again. And I think that's not uncommon. And the question is, can you explain why this works?
So you were correct. That's not uncommon. One way this is described as orthostatic intolerance, and this has been studied by several MECFS investigators such as Peter Rowe and Dr. Ilo Biagioni. And so in general, orthostatic intolerance is associated with altered regulation of heart rate and blood pressure and blood flow. And these are controlled by our autonomic and central nervous systems. So the idea here is that some of the symptoms associated with being upright are because of poor regulation at the autonomic nervous system and central nervous system level. So for example, when you're standing, you might not receive enough blood flow to your brain, and as a result, you start to feel confused and fatigued. When you lie down, you're basically helping your autonomic nervous system recover from fighting gravity and the stress of standing. And as a result, you are going to return regulation to blood flow and heart rate. You're going to get blood flow back to the brain and to the extremities, and then you'll begin to feel better.
Great, thank you. Next up, have you studied the effect on the patient of just having a conversation with another person? Can it worsen physical symptoms and show increased brain activity?
We have not studied that. In a related way, though, we are using our fatigue and cognitive tasks to see, when they're just doing a normal cognitive task, do they report a change in their symptoms versus when they perform a fatigue and cognitive task. And we're also measuring the gene expression that occurs before and after that fatigue and cognitive task. So we're going to start to begin to answer, I think the question that many have is that, is it just a physical stressor that leads to these? Because patients report that there are lots of different stressors that exacerbate their illness. What we need to know is what's the variability in that, how common is it, and what are the biological consequences of that. But to more directly answer the question, no, we haven't particularly done just having a conversation with another person.
Yeah, thank you. All right, next up, a short one. Do patients tend to get better as they age past 60 years?
Not to my knowledge. So I believe the answer to that is no. If you look at the longitudinal data, for patients with severe MECFS, only about 5% recover fully, and I think between 5 and 20% get better over time. But those who are over 60 do not tend to show a different pattern of response.
Okay. Next up, for long-term patients who cannot perform a physical exercise, can a cardiac nuclear stress test have potentially the same negative effect on symptoms as if the patient had physically exercised?
This is a great question. I don't know if that is true or not, but that would be an excellent study to perform to see if it's the physiological process of increasing heart rate and blood pressure that results in this versus some other central nervous system thing that may come on. So that would be an outstanding study to conduct, to compare, to see if you can get a similar outcome with a biological or a drug stress test versus something where the patient is physical. I don't know if you can. If I don't know what the answer would be, but that would be an excellent study.
Yeah. Okay. Can patients who are doing well slowly wean themselves off their medications and continue to feel good? Most patients stay on these meds for life. Is that true? And are there problems with long-term use of these meds? Very general question.
I am in no position to answer. I'm not a physician. I can't really answer that question. I don't know the long-term trajectory of medication use in patients, nor whether or not there is a long-term negative consequence to being on these medications. I'm sorry, I'm just not capable of answering that.
Yeah, fair enough. Question, how can patients join in the studies of our illness?
That is a fantastic question. There are several ways to join. I guess one of the first ways would be to to volunteer for the SMCI database. So you guys have a patient registry that you're creating that would allow patients to participate in research. We've been trying for years to create a national patient registry. We have not been successful yet. You can also search the NIH reporter database for funded MECFS studies and volunteer that way. You can go on the clinicaltrials.gov network and look for trials that are being conducted in MECFS. And there are many centers across the country that are also looking for patients. So if you can find the centers, you could volunteer that way. We have not cracked that nut on how to best get as many patients as we can. And that is really one of our biggest challenges as well, is to get enough volunteers to be able to conduct really meaningful research.
Yeah, everyone struggles with that. And of course, you're referring to the Solve MECFS biobank and patient registry, and we do offer that up as a service to any credible researchers who are doing work in ME. So if you are interested, you can go to our website and sign up. How did you get interested, Dr. Cook, in this kind of research, and what was your motivation?
So my initial interest was really in, I was studying exercise and pain research, and I've always been interested in the brain, so I did a postdoc in neuroscience that, Carol, you were kind enough to mention in the introduction. And when I was during my postdoc in neuroscience, it was with Dr. Benjamin Nadelson, who at the time had a CFS center, and he got me really interested in studying MECFS in addition to studying fibromyalgia. And once I began studying it, I initially took an exercise science perspective, then added in a neuroscience perspective, and then the more I studied the illness, the more fascinated I became with trying to solve this puzzle. I took a break from it when I came here to Wisconsin, and then CFIDS, it was at the time, approached me and kind of got me re-energized to study MECFS, and the study that I reported from my master's student was my re-entry into MECFS research, and now I'm back and I'm really interested in it again. I just think it's a really fascinating puzzle from a scientific perspective, and not to be insensitive to the debilitating nature of the illness, but I think it's something that we need to take a look at from several different perspectives and get a lot of experts involved, and that really motivates me to continue to study it.
Hear, hear. Great to hear that. Next question, Dr. Cook. Do you measure sleep/rest times for patients before and after exercise testing?
We measure sleep quality, but we don't directly measure yet. We don't directly measure sleep in terms of looking at polysomnography or having them wear something on their wrist. So we ask about their sleep quality with validated questionnaires, but we're not directly measuring their sleep. And that is something that, again, that's another system or outcome that we'd love to add to the puzzle. If we could also add that into and add the gut into our, and continue to look at all these different systems, I think we're going to know more than we currently do. That's a great question.
Yeah. Well, here's another great one. Are you aware of vagus nerve theories? And if so, any thoughts about that?
Yeah, so for a long time, the vagal system has been talked about in terms of pain regulation, and recently they've talked about how the vagus nerve is exposed in our peripheral nervous system to lots of pathogens, and that this exposure to that makes it a sensitive target for inducing symptoms, and having a dysregulated vagus nerve system is one hypothesis of how MECFS is generated, how its symptoms are generated. I don't know of any current research that's directly looking at the vagus nervous system in MECFS, but that's the hypothesis behind it. And this, our vagus nerve goes into many of the brain areas that we're interested in. It connects with those that we're interested in, and how centrally this illness is impacting the patient.
Thank you. Here's a question I think many of us have wondered about. What effect does CFS have on life expectancy?
Oh, that's a good question, and I don't know the exact answer. I don't believe we have great mortality data in terms of longevity. I don't know if we have a population-level database that would tell us that yet. I know it's being tracked currently, but I do not believe that we have the mortality data as of yet.
Yeah, I think that's right. I'm not familiar with ever having seen studies in that arena either. Okay, another question then. What types of metadata do you collect from patients during your study? For example, what they ate, if they'd showered.
So we collect probably lots of things that would be included as metadata. We probably don't ask them if they've showered. We do try to in some of our previous research we did track their diet a bit, but we haven't done diet diaries yet, which is an interesting thing to consider. What other pieces? We have lots of stuff in our screens and in our when we ask them about their experience in the study that we collect that we could dig up. I'd have to check about when we interview them after they're done. It would be interesting to see some of the things that they report that we could include as metadata.
Thank you. Okay, next up, have you done complete panel viral testing for HHV6, 7, or 8 on your patients?
We did that, I believe, in the first study. We did not do that in our follow-up study, and they were negative in our first study.
Okay, interesting. Next up, have there been similarities in results in chronic Lyme patients?
I'm not aware of any research that does what we do in chronic Lyme, although the symptom reports and the trajectory of the illness is similar in chronic Lyme as MECFS, but I don't think that they're doing the types of research that at least my lab is currently doing. So I'm unaware of any of those types of responses. I believe they've done some cognitive testing in chronic Lyme. I don't know of any gene expression or functional neuroimaging in chronic Lyme. Doesn't mean it doesn't exist, though.
Yeah. Boy, here's the $60 million question. So from your study, question, what did the 5% who fully recovered do to get there?
Oh, well, none of that's none of my research. So that's just longitudinal data having large groups of patients report over time. And then estimating that percentage. So I don't believe I've ever had a patient in my study that is recovered. If they have, they haven't called me to tell me about it. So yeah, so that was if I confuse that issue, the recovery data is based on research that is not my own.
All right. Well, thanks for clarifying that. And then I do think this is the last question we have time for. Do you collaborate with other researchers from other countries?
From other countries, not currently. I do in terms of, being on panels and participating in research discussions, but not on an actual study.
Okay. Ooh, hey, here's a question that just came in. I've got to squeeze in. Last question, really, this time. Usually, patients are encouraged to exercise as much, quote, as much as they can tolerate.
Right.
Does your study suggest that perhaps patients should avoid exercise?
My study says that the intensity that we use for the majority of our sample resulted in a worsening of their symptoms. So that majority should avoid that intensity. We have really no idea of the types of activities or the amount and intensity that patients can tolerate. It's so individualized. And the best advice that I can give now is that for your given situation and with consultation with your physician, find out what you're capable of doing and what you're not. And this is not just as guesswork. This is taking it very slow and doing a detailed documentation of your responses to even light level activity first. And if that results in a worsening of your symptoms, then maybe physical activity is not prudent at that time. We do not have enough information to say one way or the other that yes, every patient should try to be active, or no, every patient should avoid activity.
Yeah, yeah. Well, listen, thank you so much, Dr. Cook. You know, first off, you answered every single question we were able to throw at you, and I think that demonstrates your depth of knowledge about this illness, and I'm very grateful. But I would also just comment, you know, you are one of really a handful of researchers in MECFS who step out of the lab and take a role in public policy, and the time that you commit to serving on CIFSAC and being involved in other issues related to this illness that are very important at this time is really commendable. So thank you so much for all you do for MECFS patients.
That's very, very kind of you, Carol. I appreciate that very much. And let me say that those questions are challenging, and they're a very direct reflection of how informed this audience is. And you're taking me to the edge of my knowledge.
Well, thank you for going to the edge with us today. So with that, we will wrap up. For our many listeners still on the call, the last webinar we'll be doing for our 2015 season will be on Thursday, December 17th, with our own Dr. Zahir Nali. He's our Vice President for Research and Development here at the Solve MECF Initiative, and he'll be doing a year-end 2015 sort of a research roundup. So we do hope you'll join us for that. Again, thank you so much for listening in. We hope these webinars are informative and appreciate your attention. Take care, everyone, and goodbye.
Thank you, everyone.